me the opportunity to become the chairman of this last session of this very nice workshop. And it's a great pleasure to introduce Jean Clairambault, who I have known for a long time. And actually, last time I saw him physically was in Moscow, which is interesting, actually. So it's my great pleasure to let him give the talk on a very nice subject, which is with the title "From Mathematical Modeling of Cancer Cell Plasticity to Philosophy of Cancer," which sounds promising. So I'll let you talk, Jean.
Jean, I'm sorry, could you make full screen, please?
Sure, but I cannot share my screen. It's written, you can share your video after sharing your screen. So is it okay this way? I will now try
Okay. We can see it, so please don't change if there is a problem. Just okay. Now we have the full screen.
It's perfect.
Okay.
Yeah.
So and you can hear me. You can see the screen is perfect.
Everything is nice. I'll let you know if there's a problem.
Thank you, Laurent. Thanks to the organizers, in particular Vitaly, for inviting me to give this talk, which I may have given in different forms already, but it's always different, I think. And this time I have decided to put it under the auspices of what I call now philosophy. I'm not a philosopher by any way, but I discovered that philosophers of science who are interested in cancer, in the biology of cancer, now call this discipline philosophy of cancer. So I think that starting from mathematics and modeling, in particular, due to the questions risen by the resistance, drug resistance of cancer cell populations that lead, unfortunately, to many deaths of patients, it's a natural way to come to the question of plasticity of cancer cell populations, of cancer cells and of cancer cell populations. Plasticity, as you will see, means that you start from an isogenic, that is a population of cells that share exactly the same genome, but with different phenotypes. And they may share these phenotypes and distribute them so as to do some kind of bet hedging. And this kind of bet hedging, also I am trying with young students to model, and I'm not sure I will have the time to mention, or maybe very briefly, what we are doing with a PhD student now about bet hedging in cancer, which is related also to the emergence of multicellularity. So it's a joint work. In particular, here I was speaking of Frank Alvarez Borges, and with many colleagues of the Laboratoire Jacques Cruillons in Sorbonne University. Rebecca Chisholm and Thomas Lorenzi were postdocs. Alexander Lortz was an associate professor. Benoît Pertam is a full professor, so is Emmanuel Trella. In fact, they are the last two heads of the lab. And Camille Pouchol was my PhD student together with Emmanuel Trella, and I will present a lot of his PhD works. So, what is cancer? Cancer is certainly a puzzle, and it has been so for thousands of years. But what must be considered when you speak of cancer, it's that searching for the causes of cancer in single cells may lead you, of course, to find different things, different anomalies, but the determinants of cancer are not to be found in a single cell, because cancer is always a disease of multicellular organisms, and in fact of metazoa, that is, of animals, because even if it exists in plants, plants know how to deal with it and put it completely under control by stopping it and leave it leaving it, let's say, in a sort of by palisades, by things like that, in very localized settings, and not invading the organism, because resistance to treatments in cancer is one cause, and of course the other cause is metastases that lead patients to this. But first, cancer is a localized loss of coherence between cells and tissues. It's always starting from some points in the organism. And I contend that it begins by loss of control on cell differentiations, and then only to enhanced proliferation of local enhanced proliferations of cancer cells. In fact, if you're aware of the works of Marta Bertolazzo, who has called her first book Philosophy of Cancer, she contends that it's a two-player game. Loss of control on cell differentiations and on proliferations also. But I propose that it all begins by loss of control of cell differentiations. And this may be better understood within the framework of the atavistic hypothesis of cancer, that proposes an evolutionary origin for cancer cell populations, and I will briefly mention this. In fact, it says that cancer is a reverse evolution towards unicellularity, or towards very coarsely organized multicellularity. And cancer in this setting, in this explanation, is always trying to reinventing the wheel. But of course, it never works. In the time scale of one human life, it cannot work. And this has been documented by different teams of biologists. In particular, phylostratigraphic analysis have shown that these genes that are altered in cancer are also those that are associated with multicellularity, development of multicellularity. So these are works by people named Domezat-Lochon and Tautz, but it has also been developed by teams of geneticists in Australia. The first name is Anna Trigus. So if cancer is a disorganization by loss of coherence between cells and tissues, what is coherence and cohesion within and between cells and tissues, and why and how is it disrupted in cancer? These are my starting points. Oh, sorry, maybe I was very, very short in yes. So the plan of the talk is about facts about cell plasticity in cancer. Then I will speak about how one can model cell population plasticity by structured equations, and what are sources of plasticity in cancer. And what is a multicellular organism will be my fourth section, and how is it deunified. By the way, this is a proposal by Thomas Pradeux, who is a philosopher of cancer, philosopher of immunology, in fact, who says that cancer is a deunification of the organism. And finally, I will end by possible consequences for therapeutics in oncology. I've just checked that yes, I had forgotten a few things. So to come back to my motivations, you will see that mechanisms of the great evolution that are in fact addressed by the atavistic theory of cancer may explain transient or established drug resistance, and also at the time scale of human life, as I said, or human disease, you will see that this may be represented by reverse evolution towards, as I said, towards unicellularity. You can see intratumor heterogeneity, but heterogeneity towards what? This is a question that I will try to address. And plasticity in cancer cells, as I see it, as I will present it, is a kind of propension to reversal to a stem-like or stem-like dedifferentiated status, but it may also result in transdifferentiations, as I will illustrate later. So this plasticity makes cancer cells that are in fact cells endowed with our genome, but in this genome, many genes have been silenced, and the cancer cells are able to re-express these genes and make them amenable to resist abrupt drug insults by response to extreme cellular stress. So this reversible plasticity can be captured by mathematical models, and these models are compatible with optimal control methods, as I will present later. So did I forget? Yes, I think that I've said most of this. Now, facts about plasticity in cancer. These are the major concerns in oncology: drug resistance and metastasis. As I said, lately, it is these concerns that lead, when they are not faced, patients to death. And one can see established drug resistance as an evolutionary rescue in terms of evolutionary ecology. And I will try to illustrate what is this. We have also, and this came only lately in biology, experimental evidence of cell plasticity in cancer. I say only lately because most of the time, before, let's say, 10 or 15 years, biologists thought that cancer came only by mutations. But even if it is by mutations, in many cases, mutations towards epigenetic enzymes, in fact, what leads the evolution to cancer is at first due to plasticity, and it's an epigenetic phenotypic evolution. And this remains a big question. What is a part of genetic mutations? What is a part of phenotypic switch? So I think I said most of this, but I must insist on the fact that the unwanted side effects of drug treatment on healthy tissues and on cancer tissues must be taken into account in a therapeutic optimization perspective, because this is a practical objective that leads us to a practical modeling going towards the clinic, even if clinicians are sometimes, very often, dubitative about the utility of our models. But I'm not completely forsaking the goal to convince them. And it's important also to say that in healthy cell populations, plasticity is not present except in very special cases due to, for instance, wound healing, or in different animals like axolotl, repair of missing limbs. But this is exceptional. In principle, healthy tissues are not able to plasticity, to develop plasticity, whereas cancer cell populations can, and do it very often. Well, we can use different words, in fact, about drug resistance. Drug resistance, in fact, is usually used in the sense that it's established by mutations. And about cancer cell plasticity, that is without mutations, we most often speak about persistence or tolerance to drug treatments, which is by nature reversible. Metastasis, by the way, that I recently spoke about, is a manifestation of plasticity, because they always rely on epithelial to mesenchymal transition and its reversion, mesenchymal to epithelial transition, which makes epithelial cells, like for instance in the gut, able to become fibroblast-like cells and migrate and build metastases. And when they have established, then they can develop again an epithelial-like phenotype, but of course transformed and develop uncontrolled proliferation in metastases. And here, this is a thing we are working on with, in particular, Camille Pouchol and Nastasia Poradier-Dutey and their PhD student about an interesting thing about hysteresis and tristable multistability that has been observed about EMT. But I will not say more than this. So evolutionary rescue can be seen in cancer treatment in this, for instance, in this celebrated figure that came from a paper in Nature in 2012, which tells you that after an important amount of chemotherapy here, then the whole cell clones or subclones shrink, but after some time, with the same chemotherapy being maintained, there's an escape, what ecologists call an evolutionary rescue. And these cancer cell population regrow. Some have been, for instance, the purple clone here has gone extinct, but others can grow up, and even others can show up. So this results in what is observed as enhanced heterogeneity with enhanced proliferation and proliferating clonal cell populations. But is it always mutations? In this example, it's clonal mutations, but it's not always the case, as I will try to show you. There's been, after this observation that stimulated us with, in particular, Rebecca Chisholm and Thomas Lorenzi in Laboratoire Jacques Williams to develop models, there has been this very striking examples of an aggressive population of cancer cell, non-small cell lung cancer, and exposed to high doses of drugs that led the population here in the Petri dish to go extinct, but not completely. It is a little less than 1% of them survive. In this maintained drug bath, that is a very hostile drug environment, and these are called drug-tolerant persisters. These are these subpopulation, very tiny subpopulations that in the first stage just survive. But after some time, and it takes time really, these subclones that have survived also begin to proliferate again. So they are what these observers call drug-tolerant expanded persisters. In fact, these words come from bacteriology, where this can be observed also. And by the way, this leads us to mention to you that in principle next year in June, there will be colloquium, a symposium in Croatia about persister cells. It will be mainly dedicated to biology, but I hope that some mathematical modeling observations and proposals will come also. What is important also to say about this evolution of cancer cell populations is that there is total reversibility to the initial stage, to the initial setting, if you withdraw the drug bath. That is, you rinse out the drug in the Petri dish, and what you can see is that the proliferation starts again, and these cells do not show resistance. After they have been allowed to take some time, they are sensitive again, completely sensitive, and you can start the cycle again. The sensitivity, DTPs and DTPs, which means that these DTEPs, in fact, must show very consuming, energy consuming phenotype to face the drug insult. And if there's no drug insult, well, they come back to their peaceful setting again, and are very shortly after that, or not so shortly, sorry, if you can see here, the doubling times, it's not so short. They are sensitive to the drug. And what is also interesting is that there's a hint that it's by no means due to genetic mutations, because inhibition of the epigenetic enzyme KDM5A completely blocks the emergence of DTPs. So it seems to be a phenomenon of epigenetic origin. So this should be modeled. And in this way, I will show you how we try to model this. But about plasticity, I mentioned EMT and drug persistence, and there's, as I said, vocabulary disputes between these terms, persistence, tolerance, and resistance. But in the sequel, I will use the word tolerance to name all these either reversible or irreversible phenomena, for the sake of simplicity. And also it's noteworthy that it has been mentioned that it's possible to go from epigenetic modifications towards mutations, and of course the reverse is true by mutations of epigenetic control enzyme genes. So it's not always so clear that it's due to epigenetics or to genetics, but we took the bet that we could represent the epigenetic way of functioning, of representing this phenomenon. Do I forget? No. So now about modeling. We have to model adaptive mechanisms, adaptive and reversible, and by PDEs or by integral differential equations to represent drug persistence or tolerance. These equations are in fact phenotype structured by continuous phenotypes, as I will show you, in cell population dynamic models. And in adaptive dynamics, you are concerned with the evolution of the trait, of the probabilistic distribution of the traits, and also on the asymptotic behavior of the cell population, be it in terms of total populations or in terms of description of the phenotype that structures the population. When we say structured population, well, we mentioned biological variability seen from the point of view of what is important to describe this variability. And for us, in what I will present next, it will be expression of drug resistance genes. And then optimal control therapeutic strategy I will present to you. So the first model that we used, that we proposed, and that was published in Cancer Research in 2015, was due to a question I proposed to Rebecca Chisholm and Tommaso Lorenzi to explain this two-stage progression of the phenotypes. And I came to the idea that there should be two different phenotypes. And for me, it was expression level of survival potential phenotype and expression level of preferential potential phenotype, which later in different talks and papers, I discovered that theoretical ecologists call viability and fecundity. But I did not know these words when we began to, with Tommaso and Rebecca, to represent by this ad hoc model the evolution towards drug tolerance and or persistence. So it's a reaction-advection model with stress-induced adaptation. This is where the stress, the cellular stress, is represented, and it's dependent on the drug. Whereas this term, the reaction term, is a kind of a non-local Lotka-Volterra. Non-local Lotka-Volterra because here, this proliferation term is in fact of the form, as you can see here, a proliferation term, A1 plus etc. 1 minus X, multiplied by 1 minus rho over K. And rho is the total cell population. So it's Lotka-Volterra-like, and it's non-local. And we also added, it may be by a kernel integral, but for simplicity, it was maybe too simplicist, but it was represented by a Laplacian. So you have non-genetic phenotype instability, which is supposed to be of epigenetic nature, the term of selection, and the term of adaptation. And here, I will not give you all the results, but it was dealt with simultaneously by an ABM, an agent-based model, by Rebecca, and by a PD model, the previous year I showed you, by Tommaso. And they obtained convergence results that I will not show you, but you can go to the paper of 2015. So after this ad hoc model, with Tommaso beginning and then with the beginning thesis of Camille Pouchol, we decided to propose a general setting of what should be a phenotype structured population dynamics model. So it's correspond to the description of evolution of a population in time t and in a relevant phenotype. This X phenotype in this slide is represented as a one-dimensional one. It's continuous in our way of thinking, but it may be multi-dimensional, of course. And in the previous example, you saw that it was two-dimensional. And in other settings also, it may be two-dimensional with constraints imposed on the behaviors of the two, for instance, traits that may be identified as viability and prolificity, or prolificity and motility in other settings. So here in this simple setting, you have the one-dimensional trait, and you can follow either the evolution of individuals making the population, that is evolution of tumor growth, and the asymptotics, or simultaneously, of course, you do this, of distribution of the trait in the population. Here, this is obviously a probability measure. So tumor growth, asymptotic distribution of traits. So first question, what is the asymptotic behavior of the total population? And second question, what is the evolution of the phenotypes in the population and their asymptotic distribution? So we exemplified this model by, again, a non-Lotka-Volterra model with a simple idea that there's a trait-dependent instantaneous proliferation rate tempered by, hindered by a non-local logistic term here, where the depth term may or may not depend on the trait also. And so this was a simple model from which one can easily see with a simple hypothesis that in this simple case, you have always convergence, non-monotone usually, non-monotonous convergence, but convergence to a plateau. That is, the total population goes to a plateau. In this setting, remember the logistic term, of course, it does not go, how can I do this? Please, can I, well, never mind, I hope it will not be the same. It will go to a constant which may be identified as a maximum of the terms rho over D. So now, what can you say about concentration? I make a difference between convergence with respect to time and concentration with respect to the traits. Well, here this is a succession of snapshots of a film, in fact, where you can see the plot of the distribution of the traits for different times T of the population. So here you start at T equals 0 and you finish with the Dirac at T equal 100. So the theorem states that you have convergence and concentrations of the population on a Dirac-like set. If you had the hypothesis, which is not given, that this set is reduced to a singleton, to one or only one element in this set, then the total population's convergence in this measure space towards a Dirac with a total mass rho infinity, the plateau that I showed you. Now, in another maybe more complex setting, that is without just using the fact that BV hypothesis is enough, Camille Pouchard proposed that by using this Lyapunov function, in fact, you can present the idea that you have at the same time, and this will be used when there are more than only one cell population, you can show that the derivative of this Lyapunov functional consists of two parts with the same negative non-positive sign that shows independently or separately the fact that rho will converge to a plateau, and that this integral going to 0, well, this set that bear that supports the X phenotype is of 0 measure. So this is, of course, an added hypothesis. If this 0 measure set is a singleton, then you have convergence and concentration at the same time. And this Camille Pouchard will use in the next thing that I will show you about not this part, which is biological again, but in the application to optimal control in cancer. So again, a bit of biology. What is plasticity? It is, of course, always epigenetically determined, because I remind you that it relates to phenotype evolution in isogenic populations. And isogenic populations, of course, evolve to make such complex systems as we are, that is animal multicellularity organisms, but this is only possible by epigenetic modification, that is by the influence of epigenetic enzymes and by changing phenotypes, but not genotypes. And this possibility of de-differentiation, that is one manifestation of plasticity, may also be accompanied by, in other settings, or in the same settings as I will try to sketch, by transdifferentiation plasticity. Well, this is in particular illustrated by this different phenomenon. De-differentiation may be present, for instance, in acute myeloid leukemia, where there is blockage of differentiation. But there can be also uncontrolled differentiation with transdifferentiation, leading to immature cell proliferation in diseases such like Barrett's esophagus or ductal carcinoma in situ. Transdifferentiation also may be illustrated in other settings that are very rather strange, in fact, and for instance, trans differentiations of epithelial cells leading to a neuroendocrine serotype. This is typically trans differentiations. And all these are manifestations of plasticity. I'm just saying in a few words that if you think of cancer stem cells as those that are atop of the tree of differentiations, well, they are not always needed. Cancer come without from cancer cell modifications, mutations, but not only because, for instance, of what I mentioned about the AML, AML3, in fact, acute, what is it? It's a transient stage of differentiation that blocks differentiation, and it's a kind of acute myelogenic leukemia that can be totally cured by drugs that have been found in the last 20 or 30 years. So differentiation control may be altered at any differentiation stage, and it would be nice to find at what stage has been, this is promyelocytic, sorry for the word that escaped my mind. It would be nice to find at what stage this has been stopped, blocked. Also another...
You have 5 minutes.
5
left,
minutes, okay.
Jean.
So I will not mention this stress launch dedifferentiation signals. I will just mention, if you're interested in this, the paper in PNAS 2015, and the fact that it refers us to the so-called atavistic theory of cancer by Davis, Lineweaver, and Vincent. I will show you this in a very accelerated way. So the atavistic theory of cancer just states that it's a reverse. Cancer is a reverse evolution from our evolved mammalian capability towards, not until, but towards earliest evolved capability common to the last uniform, not uniform, unique common ancestor. And in this way, cancer is more an archaeoplasm than a neoplasm. And the atavistic theory of cancer also gives us a hint that it's a reversal at this time of the evolution of our planet between 1 billion of years until 500 million years of the oxygenation of oceans in the atmosphere. And this multi-cellularity toolkit that was developed at that time should be documented. And this has been, as I mentioned, done by Domazet-Locher and Tautz and by Triggers. And well, attacking cancer on proliferation and where it is most powerful is not the best thing to do. Another evolutionary framework is, of course, the epigenetic Waddington landscape that many of you, I suppose, know of, and it has been mentioned also by Sui Huang, who is a systems biologist, saying that these bifurcations can be illustrated by stability phenomena that can be represented by ODEs. I have also another metaphor to propose. These were metaphors, the previous one, which is what I call the wicker-work basket, where evolution of the construction of multicellularity starts from a base, is here, and is constituted of fibers that are also maintained together by horizontal cohesion mechanisms that are not cellular, but likely of metabolic gene regulatory networks nature. And this makes the construction of the multicellularity organism until they evolve the rim of completely differentiated setup. I am working with a colleague to try to mathematize this, but this is not completely easy. So possible consequences for therapeutics, and this is where mathematics is coming back. There is certainly insufficiency of single cell-based therapy, and this is where the center of my talk should be located here. We have this two population, non-local Lotka-Volterra-like model with the inclusion of the logistic term here of a non-local nature. Here it's a compound of the healthy cell population and of the cancer cell population, rho H and rho C. And there are also terms for targeting the death term and the proliferation terms here. And the question that is addressed by this optimal control problem is associating in an optimal way these two drugs, cytotoxic that addresses the death term and cytostatic that addresses the proliferation terms, with additional constraints, state constraints, in fact, present at each time. And this has been addressed by Camille Puchol in his thesis, following the same idea of the Lyapunov functional, but showing that you can combine two Lyapunov functionals to show that exactly the same result hold. That is, you have both convergence and concentration exactly at the same as the same result were presented, but for the two populations. And this setting has the interesting property that it can show you what results from a constant and high dose of drug in cancer. You certainly will have first diminution, decrease of the cancer cell population, but quite soon you will have concentration on a resistant phenotype. This is the peak that you can see here, and with catastrophic effects on the healthy cell population and on the cancer cell population alike. So the optimal control problem has been stated by this theorem in Puchol et al. Paper in Journal de Mathématiques Pure et Appliquée. The proof uses a Pontryagin maximum principle, and it tells you that there's bang-bang, free arc, and bang-bang again control. And this can be illustrated by this illustration of the APM IP-opt algorithm that states you that here you must give nothing in the, or almost nothing in the cytotoxic drug flow, giving a very short non-singular, singular arc for a very short time, and then again bang-bang control at an admissible level. And you're freer for the cytostatic. But what you can see is that after the slow growth of the tumor cell population, there's a going very fast decrease towards extinction, whereas the healthy cell population is preserved at the prescribed level. So also we must have in mind immunotherapies, which is still a cell-killing therapy, but, and I have been beginning to design such models, but they are just toddlers now. It's a reinforcement of the killing power of the immune cell police. It's not the non-cell killing therapy, which I call with my wishes, but it's not exactly for the present. It's a future prospect, and it's what I call reformatting the cohesion watch. That is, redesigning the likely metabolic gene regulatory networks that control the compatibility and the cooperativity of tissues and cells that make a multicellular organism. This cohesion watch should be identified. I mentioned these metabolic gene regulatory networks which in fact was an idea that came to me while listening to a proposal by Vitaly lately. But there's much to be discovered. And this should induce us to find new way to enhance these mechanisms, but not necessarily with cell-killing therapies. And so, as a conclusion, I will say that we may use certainly new therapeutic tracks with old drugs, but by using optimal control, but you have to optimize these combination therapies. What is also bearing many hopes are immunotherapies, and we have still much to understand when we think modestly that William Colley's founding experiments in cancer immunotherapy have not yet been completely explained. So these about reestablishing the cohesion watch may be considered as mere speculations. Well, I'm not so certain about this. If we can have these things in mind, then we can be sensitive to observations and possibility to lead other optimal control settings to reestablish a coherent and cohesive multicellular organisms. These are a few references. There have been also a few after this one, but finally, I thank you very much for your attention.
Many thanks, Jean. We have like maybe time for one or two questions, no more. So if you have like one question, yes, I think it's Vitaly. It's written in.
Yes, thanks. Thank you, Jean, for this really important review of this new insight in the cancer theory. Unfortunately, we don't have time to discuss it in detail now. We had several previous discussions, but I continue to learn about it. And very short question now. Of course, we know already about epigenetic mutations and their differentiation, and you explained about the atavistic theory of cancer, but all of them do not imply genetic mutations. On the other hand, this is my question, are there examples of cancer, of tumor growth without genetic mutations?
I cannot be completely negative nor positive. In fact, I think that experiments by, for instance, Anna Soto and Carlos Sonnenschein have shown that cancer cell populations put in physiological natural settings can revert to normal cell populations. So what does it, how can this be interpreted? Well, maybe there were no mutations, but maybe also there were mutations, and these mutations, these mutated cells have gone extinct in unfavorable settings. But what I'm also I have also presented is the idea that cancer may start from mutations, but not directly on the cell populations, cell being mutated, but on mutations of epigenetic enzymes, and sometimes it may take quite a long time for these mutations on epigenetic enzyme control genes to reach the whole cell populations. So I think that the scenery is not completely clear. Do we have a completely, is there a possibility to have cancer without mutations? Well, I doubt it, but the mutations can be very tiny and very discrete and may lead to cancer. For instance, in leukemia, this has been documented, maybe 40 or 50 years after the initiation of such an epigenetic mutations. And this has been documented, as I said, because there were observations of people who were perfectly people victims of road accidents who are perfectly healthy, but you could see in samples of their blood that they already had mutations of at these epigenetic genes locations. So it can take much time, but these epigenetic mutations may be also always, I cannot be completely positive, but can be always present at the very onset of cancer. But can it be completely excluded also that cancer may be without mutations? Well, there is also this hint that I mentioned to you in one of my slides. It's possible that mere phenotypic changes may force by stress cell populations to mutate. So there can be cancer without initial mutations, but I think that there's always somewhere a hidden mutation. But if you wait for this mutation to appear, well, you can miss the target. And I think that in the representation of drug resistance or drug tolerance, it's more important to address the plasticity of cancer cells. That is obviously of epigenetic nature.
Okay, Jean, thank you very much. It's really very important question. Thanks.
Thank you. We can, I guess we don't have time for the next question. Sorry, because we are like 6 minutes late. So we're going to, thank you again, Jean. It was really interesting. I have like a lot of questions too, but we don't have time. Let's switch to the next speaker for the next session. So you can unshare your screen, Jean,
Yes,
I think.
this is.
I guess, yes, thank you. And let's get the opportunity to Alexander with nice hippopotamus picture to share his screen. And if you can switch to the full screen, I think it should be working. Oh. Okay. Okay. Looks good. So Alexander, it's a great pleasure for me to introduce you as the last speaker of this nice workshop. So you're going to talk about the mathematical model of pancreatic cancer, and I give you your time for speaking. Thank you.
Oh, maybe I will continue the idea that was developed in previous presentation in some concrete case. It is a pancreatic cancer. And next, how's next? One moment. Some difficulties.
Usually you can press the arrow on the keyboard. It's not working. No.
Maybe I will demonstrate it with small screen.
Yes, no problem.
Not problem.
It's clear enough. Yes.
In pancreatic cancer, it is one of the difficult problems of contemporary medicine. What is the reason of this? Pancreas is arranged in stroma. It is some special sake that did difficult its inspection and prevent for obtaining therapeutic beans. In other words, sake it is an obstacle for penetration drug for pancreas. And second, there existing a very specific phenomena in a symbiotic behavior between cancer cells and pancreatic cells during long time of progression illness. It is an unusual phenomenon, but in medicine established that the phenomenon exists. And second, more important, the process of development of pancreatic cancer is presented as a chain of different, relatively rare mutation. And result of this mutation, we obtain set of pre-cancer and cancer cells. Now, for example, is the normal cells and result mutation becomes K cells, K and Kp, and go on. A classical model of tumor progression is presented here. Most mutation occurs as random, do nothing of damaged cells, because there exists some mechanism of repair cells. But a few specific mutation can make cells grow and proliferate faster and survive more, and therefore can lead to uncontrolled growth of tumor. In this slide, you will see some typical picture. We have some normal cells, and those are mutation 1, we appears as cells with star, and mutation 2 in continue, and at last we have a set of cancer and pre-cancer cells. Different normal cells types have different mechanism of growth control, different cancer causing mutation within some cells type with cause of different phenotypes. In most large aggressive tumors, it is a harbor set of many different mutations, and that not uniform. This is maybe one of the difficult problem here, because why? Main problem of chemotherapy related to this phenomena is that the treatments that decrease the birth rate only one of two kind of cancer cells. No, as usually say, is wild type, but do not decrease do not action for other cancer cells which resistant to these drugs. That some type of mutation, some type of cells as a result of mutation become resistant of those drugs. That is maybe the reason the first chemotherapy and second very successful, but after that we have no success. For description of this phenomena, we will use an eigenreplication quasi-special model. It's very briefly about this model. This model consists of two matrices. It is a matrix M, diagonal matrices. It's a so-called fitness landscape of community species. No, in our case, it's community of the cancer cells. What does mean me1, me2, me L? It is coefficient of proliferation. And second object, very important, it is a matrix Q, where Q sub EG is probability that under replication of species with number G, we appear the species with number I. In this model, P sub G of T is a frequency of the species, is a relative value. And we have some not so very difficult differential equation. This is matrix Q multiplied in matrix M that depends on fitness landscape. This is value M with line. It is so-called fitness of this system, a fitness of the system which determined of this equality. It is very good model and used in many investigation in theoretical biology, but only one lack here. There we have no death rate for the species, because we consider this model in some prime ideal bouillon, and total sum of species is equal at the same value. And for improve for this lack, we consider some modification of this model. It is so-called open quasi-species model. We presented this work when we presented this model. The Q and M are the same matrices as before, but we introduce the death rate matrices, these diagonal matrices, and all equations have this following form, where S of t is a total sum of number of species. And the fitness of this system, a little bit other, it is an old fitness is here, and in denominator we state that this value can take into account death rate. And so for model for cancer, of course, it is a very suitable model that take into account in considering this rate. No, it is continue open quasi-special model. We can investigate steady state position, and the fitness this have at the same value, and there we have one rest point only. This is limit of this value. The OT it is satisfied in previously equation. And now we consider some concrete cases, pancreatic cancer. That is a chain of sequential mutation of precancer and cancer cells. And total number of all mutation species are 64. This only one part of this mutation. It is the normal cells as a result of mutation. Very small probability in 10⁻⁷ become NK cells. After this result mutation, we can appear Kp, Kc, Ks cells, and then continue, and at last we have eight kind of cells. What is the question that arise? These cells? It is mentioned that the cancer is over, is appear or no, but it is a very difficult question, of course, because all this set of mutation cancer cells not busy give the cancer. Only some combination of these cells, it is established now exactly, give a cancer. And therefore, not simple problem. What is mean cancer in this case? This is a mathematical model of pancreatic cells. We used open quasi-species model, and this what terms very interesting. It is effect of symbiotic action between cancer cells and normal cells. Here, this is action, symbiotic action of cancer cells, and here the symbiotic action of normal cells on cancer cells. And we considered together the action of immune system. That is this T cells. This equation describes the action of T cells on the cancer cells. You can see here. U0, it is the quantity of normal cells. UE, this is for N64, it is the quantity of cancer cells. T of T, quantity immune cells. Co-mutation matrices from quasi-specimen model, NM, and fitness landscape. Some biological information, it's very interesting. What after the mutation normal cells with probability 10 minus m to appear K cells. This is a doubling days for K cells, death rate. In information we used for very important object. It is this mutation matrices. Using this information, we can construct the mutation matrices for all 64 cancer and precancer cells. Healthy cells have a doubling 6,100 days, and it has a death rate in 2%. Here's the biological information about the equation of immune systems. This table was presented to me by co-author Gennady Bocharov. And this is a very simple calculation, real-time progression. What is the time here in this process? And we calculate the coefficient of landscape, fitness landscape for normal cells. It's nearly this value, but is important for understanding what is the time, not time of abstract time, but some real time. Now I will present the result of some numerical modeling of these systems. We will see two pictures, one in left side, one is right side. On the vertical axis, mapped the values of frequency cells. It will be cells, normal cells, K cells, Kp, and many other cells. On the horizontal axis, postponed time, where 1 unit, it is equal 1 month. And graph in left side represent dynamics of frequency without action of immune system. As left side, it is this, value of frequency of the cells without action of immune system. On the right side, we shows moving dynamics with considering action of immune system, but in reverse order, from maximum to minimum. But initial data for this system is we have only normal cells without cancer and cancer, and initial data for immune system is here. You will see these pictures for normal cells. Normal cells will decrease, and without action without action of immune system, and at the same picture with action of immune systems. Here the immune systems bigger, this less, inverse order. And at the same picture, you will see for K cells, for P cells, for all kinds of these cells. Some of this picture is analogous, but some is different. I only show the picture for KPC cells, KPS cells, KSS cells, PSCs, and at last it is a finished KPSC. But all number of the picture, I remind, it is 64. This is the result of modeling graph dynamics test cells. It's a big action, influence, and then slow and slow. This is the T cells. On this picture, you will see a graph of fitness depends on the action of immune system. It is without immune systems. No, it's fitness here. It is a fitness, not good sense. It's a fitness of cancer cells, typically. And it is, we will that is increase in the very, is a limit size that is 1, but this is cancer cells, in spite of all these cancer cells have a small frequency, but in the reason that the number of these cells is 64, in total they have a 70% of the total of all this of all cells. And in the finish, I would like to say here, this is some literature you will see. I would like to say that of course the time of development of cancer cells in this process is very high. But really in medicine people told that cancers, that the pancreatic cancer can develop long time, 10, maybe 10 years. But in our calculation, we obtain little bit more period. It is this 16 years. It's the first. Second, we don't take into account the very important process of competition between different kinds of cancer cells. And we will continue this investigation and take into account competition too. And second, we don't account this systems the food, nutrition. It is very important too, but it is possible included in this element in our systems. Thank you. I'm finished.
Thank you, Alexander. Just before getting some questions, I have one. It's actually the last sentence you gave. Is like nutrition is very important. In which way? Is it specifically for the pancreatic cell cancer or for any kind of cancer?
We have some community, of course, yes, nutrition and oxygen and glucose, and therefore, if the cancer cells grows up, the nutrition for normal cells is decreased. Of course, it is very important, and important interaction between cancer cells too. It is very important things because we know that a very close mutation of cancer cells have a sufficiently big competition between
for nutrition. Okay. Thank you for your nice talk, Alexander. Do we have any questions? Yes, Vitaly.
Yeah, thank you for the presentation. As far as I understand, you mentioned in the beginning that there are many different possible mutations, and probably I conclude from that there are also possibly different types of pancreatic cancer. Is it so?
Yes.
According to different mutations, right? Or it's not a single mutation in the whole pathway of mutation. So maybe there are these different pathways, different types of cancer. So is it possible to take into account this evolution, cancer evolution, in the sense of different mutations and different subtypes?
Yes, of course it is possible, and we did there, but another model. We introduced sometime time of evolution. It is more slowly time, that time of action, real action of the system, time of evolution. And if we introduce this time of evolution, it is possible to present all the same equation in steady state position, when all element of the steady state position depends on the evolution time. But what is the purpose of evolution? Purpose of evolution, it is this increasing, according to main theorem of Fisher theorem, it is increase of fitness of the system. And possible to consider this process for aim of increase of total fitness of all systems. And very interesting, so this model, it is possible to understand how the increasing of death rate of one type of cells, action of all the process. Usually, if you have some, for example, wild type уничтожается element very strong, on the happening some change on the place of wild type, go other cells that before the evolution have no so big role. It is impossible to do. Mm-hmm.
Thank you very much. So the unfortunate conclusion is that cancer increases the fitness of the population. This is very bad for us. Okay, thank you very much again.
Thank you. If there is no more questions, I think I can conclude these sessions and let Vitaly talk about the closings. I mean, closing talk and some general announcement.
Thank you, Laurent, and I would like to thank the plenary speaker, Alexander, for the session, and also Laurent, our chairman. And yes, then we will start our closing session. And in fact, first of all, in this closing session, I would like to thank the organizers who helped us to prepare this workshop. I will name all people involved: Anastasia Mazokhina, Alexei Tokarev, Christina Leon, also people who helped us as technical moderators. Again, Christina Leon, Christina Lumina, Lilia Parashutinskaya, and Viktor Kruglov, and also the Roden University for the technical support. Okay, and then we have, in fact, two questions during this closing session. We are going to present some journals where the participants might be interested to submit their papers, and also, as suggested by some participants, to present shortly some educational programs, in particular master programs. And I would like to invite Yuri Vasilevsky, I think, here. Yuri, do you hear me? I give you the right as a speaker, so if you want to show your slides.
Yes, I hear. Hello, everybody. First of all, I want to thank Vitaly for his efforts to organize this workshop and for the synergy he promoted to us. So I think that we all are very grateful to Vitaly for his wonderful.
Thank you very much for this nice words, and it was a pleasure for me to invite all these friends and collaborators. So, big pleasure.
Yes, and now I will give a small presentation about journals which I want to promote. Do you see my slides?
Yes, yes, we do.
Yes.
Okay, so I'm going to talk briefly about 3 journals and 3 magister programs, which I want to say what I want to say about. So the first journal is International Journal for Numerical Methods in Biomedical Engineering. This journal has more than 10 years history, and it is in the first quartile. Sometimes it goes to the second quartile, but usually it is in the first quartile. It published pretty good papers in numerical methods and mathematical models. And what is the scope of the journal is right here. So models and applications in biomedical engineering. This journal tried to follow the tactics of 2 months till the first decision whether the paper will be published or not. Of course, it takes more time to publish it because of reviewers, of several circles of reviewing, but in general, it is quite fast, and there is no fee for publication. The second journal is Russian Journal of Numerical Analysis and Mathematical Modeling. The scope of the journal that it published Russian papers on theoretical aspects of numerical analysis and applications of mathematical methods to simulation and modeling. The crucial point that at least one of co-authors of the paper should be Russian. That's why it is written new original Russian papers. So it takes every 5th issue of the year, we have a special issue dedicated to simulation in biomedicine. And these special issues publishes papers as proceedings of Biomath conference, which holds every year in the beginning of November, and the next the 13th conference will be this November 2nd and the 3rd November. People are invited to look at the program, and it will be online, and join us if they want. Also, the journal tries to keep the strategy of 2-3 months to first decision on paper acceptance or not. no fee. This is second quartile, and it is published in Germany. The third journal is Cardiovascular Engineering and Technology. This journal is on the border between second and the third quartile, and the scope of the journal are techniques which are shown on the right of my slide, and they publish mathematical models as well, and they are also quick to the first decision, about 2 months. No fee as well. So these are 3 journals which I would recommend you to submit if you want to publish your papers in English. Next, my small promotion of educational programs, in particular master's programs. So I would say about 3 programs in Russia. The first program is in Moscow Institute of Physics and Technology. This program is not only master, it also prepares, educates bachelors. And this program is organized by the Chair of Computational Technologies and Modeling in Geophysics and Biomathematics. By the way, this is pretty popular chair in the mathematical faculty of Applied Mathematics, Faculty of Applied Mathematics in Moscow Institute of Physics and Technology. The second university is Sechenov University. This is the oldest, the largest, the richest university, medical university in Russia. Last year, we celebrated its 260th anniversary. And it also provides bachelor and magister programs in mechanics and mathematical modeling dedicated to biomedicine. And the last program, this is master program, which will be provided by Sirius University. This is a new university, appeared 2 years ago in Sochi. Now it is called Sirius Federal Territory. It will open its master program since September 2022. And the name of the program is Mathematical Modeling in Biomedicine and Petroleum Engineering. The program is very interesting and intensive. I know this because I am in charge of this program. That's it, what I wanted to say. Thank you very much.
Your microphone is off.
Yes, sorry for that. I'd like to thank Yuri for this presentation, and I hope that students and professors who are listening to us can promote this information around them. And also, I would like to invite Daria Neverova. Daria, do you hear us?
Yes, here, yes, hello.
Yes. Daria can present some educational programs in the Rudan University. Please, Daria, go ahead.
Yes, thank you very much. First of all, I just want to say a big thank you for this nice organization and nice workshop, and for the opportunity for me present our results here. So sorry, children want to.
I see that you have some students there.
Yes, already. So honestly say, I don't have present some good big presentation. I just want to say a couple words. So here in Rudan, in our mathematical institute, we have the master program that calls Mathematical Models in Interdisciplinary Research. And since Professor Volpert, among other things, is a director of the Interdisciplinary Research Center in our institute, he and the staff of his center actively work with students, both in the frame of scientific seminars and in classrooms. So this program is 2 years program, and the main topics that student studied there is how formulate problems, formulation of the problems, analytical and numerical techniques for solution, and the computational of useful results. So we pay attention to these things. And so maybe that's almost all. So I just want to say that we try to talk in the frame of this program about different application of mathematics. This is mathematical models in biology and medicine, models in economics and ecology, non-locally boundary value problems, special chapters of mathematical modeling, and nonlinear problems of mathematical physics, and some analytical and numerical methods of hydrodynamics problems. So something like this, and maybe if you are interested in, it would be better to contact with us using this information, this email address or QR code. Yes, so we'll be happy if you join us. Thank you.
Vitaly, your microphone is off, I guess.
Vitaly, please turn on microphone. The microphone is off.
Yes, I think Vitaly froze. His video is frozen.
Problem.
Oh.
Vitaly, I cannot hear you.
Microphone is off. Oh, yes.
Okay. Yes.
Okay.
Yes.
Now
Sorry.
it's good.
Sorry for that. Yes, Anas, I'd like to invite now Anas Bushnita. Anas, do you hear me?
Yes, I hear
Yes.
you. Yes.
Yes, please. Anas is editing a special issue, and I think might shortly present it. Please, Anas.
Thank you very much, Vitaly, and thank you again for inviting me to this workshop, and thanks to the organizing committee for making this event really interesting and wonderful. So I want to promote this research topic that I am co-editing with Professor Vitaly Volpert and Professor Gennady Bocharov. And it's about the integrative modeling approaches in mathematical biomedicine. So here the question is about all type of models that involve interactions between different physiological processes. It could be like tumor-immune interactions or fluid structure interactions, multi-scale models, PK/PD models, also multi-scale models and hybrid discrete continuous models. And there are different applications in different fields, like in epidemiology, immunology, tumor growth, oncology, and so on. And of course, it's not limited to that. It could be other problems related to physiology and biology. So we are interested in both the development of methods, their numerical analysis, their theoretical analysis, and also their applications or the developments of new methods that describe a specific problem. And so the abstract of this for this research topic is November 26, but the full manuscript will be can be received until June 24. So if you don't submit an abstract, you can still submit a full manuscript. You don't have to submit an abstract to participate in this research topic. The journal is Frontiers in Applied Mathematics. And if you are not familiar with the Frontiers journals, they are known for rapid publications, so they process the papers very fast. They have efficient review system and transparent review system, so you can track the progression of the peer review. On the other hand, they come with article processing charges. So for this journal is 1,150 for original research paper and less than that for mini reviews or perspective papers. but you could check if your institute can finance this APCs, and if you can't, or if you are if you live in country like low or middle income country, or if you can't just if you don't have the funding, then you could reach to us and we will ask the journal managers to give you a discount. So yeah, I will just leave this link in the chat, and yeah, thank you very much.
Yes, thank you, Anas, for this presentation of the special issue. Well, I will show you also a couple of journals. Just a minute, I will share my screen. Well, hopefully you see it.
Yes, we
so
see
this journal, yeah, okay, yeah, the journal Mathematical Modeling of Natural Phenomena, which I'm editing and organized, in fact, 15 years ago. So what is interesting about this journal is that it publishes papers, all papers, in fact, in open access, and these papers are free for authors. So it's usually a unique program developed by the publisher. Publisher is EDP Sciences, French publisher, a unique program which allows to publish open access papers without paying for them. And another journal I would like to shortly present on behalf of Angelique Stefanou, who participated in our workshop, but she cannot be present now. The journal is Acte Biotheoretica. This is a very good journal, in fact, one of the oldest journals in this field with a very high reputation. And there is a special issue written here, SFBT, but there is no deadline, so you can also for your submissions create this special issue. And more details you can get from Angelique Stefanou. So yes, I stopped sharing. Yes, well, and also I know that there are some other editors of the journals here, among us, Gennady Bocharov and maybe some other. So, well, I think that just participants can contact all these people in case if they have submissions. So I think that the program of our workshop is finished. I was happy to organize it, to meet all people here. I hope you enjoyed this meeting and all these presentations. And I'm sure we will certainly continue these meetings. It's much easier now to organize them. So it's not, the next meeting is not fixed yet, but we will do it. So thank you very much for coming for this workshop, for participation, and we will meet later. Thanks and bye for now.
Thank you, Vitaly.
Thank you. Thank you. Bye-bye.
Thank you very much.